Assalamualaikum and good day. My name is Farhana Adlina binti Muhammad Adnan. My matric number is 181920. My supervisor is Prof. Thomas. Today I would like to present my final year project proposal. The title given is the degradation of methylene blue using calcium peroxide. Next, we move on to the research background. Nowadays, one of the biggest concerns of environmental issues is the discharge of dye wastewater. These streams of water will affect the aquatic ecosystem in a number of ways, such as depleting the dissolved oxygen content or settlement of suspended substances in anaerobic condition. Methylene blue is commonly used during this dyeing process, and it is released in the textile effluent, which will affect the aquatic ecosystem. In order to control the environmental contamination in wastewater, calcium peroxide has been extensively used for treating surface water and groundwater due to its ability to release oxygen and hydrogen peroxide. As you can see from the pictures, calcium peroxide has been progressively applied in terms of environmental protection, such as wastewater treatment, surface water and sediment restoration, and the third one is groundwater and soil remediation. Calcium peroxide will react with water to produce hydrogen peroxide and oxygen, as well as CaOH2. The stability of calcium peroxide makes it an effective solid phase to supply hydrogen peroxide and oxygen for aerobic biodegradation and chemical degradation of contaminants in water and soil. Calcium peroxide has exerted great performance in removal of dyes, chlorinated hydrocarbons, petroleum hydrocarbons, pesticides, heavy metals, and various other contaminants. And then we move to the problem statement. In Malaysia, textile industry and dyeing mills are very famous. In particular, this industry use many kinds of artificial composite dye, and they will discharge large amounts of highly colored wastewater. Dye wastewater contains a large number of contaminants such as toxic organic residues, acids, bases, and inorganic contaminants. In order to overcome this problem, the research for environmental friendly and low-cost treatment is great importance to control the dye wastewater. There are various methods of dye wastewater treatment have been studied and developed, such as physicochemical, chemical, and biological treatments. However, they are quite expensive, rather inefficient, and have operational problems. As calcium peroxide is one of the solid peroxide that has been increasingly used in contaminated site remediation due to its ability to release oxygen, H2O2 and CaOH2, oxidative degradation of contaminants by calcium peroxide is a new method which can provide a good alternative for the decolorization and removal of dyes. The one that is in red block is the major problems that is used to treat with calcium peroxide and water. So this is my significance of study. This study can help the textile industry, especially those dealing with dye effluent, to treat wastewater more efficiently. Dye in wastewater contains high concentration of pollutants. Due to the high cost of chemical, physicochemical, and biological treatments, chemical peroxides can be used as an alternative way in degradation of methylene blue. Other than that, this study can give contribution to the existing data, especially in dye degradation for future research. sorry. This is my research objective. The first one is to investigate the performance of calcium peroxide on the degradation of methylene blue in aqueous solution. Second, to study the effect of various parameters such as the effects initial dye concentration, pH and temperature on the degradation of methylene blue. The third one is to investigate the degradation efficiency and overall removal rate of chemical oxygen demand in methylene blue aqueous solution. My scope of work are divided into three. The first one is sample preparation. Under the sample preparation, I have to prepare the methylene blue aqueous solution and also the preparation of calcium peroxide in water. The next one is laboratory testing, which is to investigate the various parameters of initial concentration of dye, pH and temperature. The last one is sample analyzing, to measure concentration using UV spectrophotometer to calculate degradation efficiency and chemical oxygen demand. So the next one is the literature reviews. Based on journal Science Direct, biodegradation such as biotic degradation and biotic decomposition is the chemical degradation of contaminants by bacteria or other biological means. Biodegradation is a key in the natural attenuation of inorganic chemical compounds of hazardous waste sites. Organic material can be degraded aerobically or anaerobically. Most biodegradation systems operate under aerobic conditions, but a system under anaerobic condition may permit microbial organisms to degrade inorganic chemical species that are otherwise non-responsive to aerobic treatment and vice versa. Biodegradation is a natural process by which spilled inorganic chemicals or other inorganic waste material can be broken down into nutrients that can be used by other organisms. The next one is the methylene blue. Methylene blue is an organic dye which has wide applications including cottons or wools dyeing, paper coloring, temporary coloring hair and coating for paper stock. It can cause human eye burns and animals. It may also stimulate the gastrointestinal tract and cause nausea, vomiting and diarrhea if ingested. Thus, it is necessary to remove methylene blue from aqueous solution. Based on the Journal of Application of Calcium Peroxide in Water and Soil Treatment, calcium peroxide or CP is a kind of white or yellowish solid which is belonging to the group of alkaline earth metal peroxide. Calcium peroxide is used in supplying oxygen for microorganisms as oxygen generator and or directly removing contaminants from the soil or water as innovative oxidants. The stability of calcium peroxide makes it an effective solid phase to supply H2O2 and oxygen for aerobic biodegradation and chemical degradation of contaminants in water and soil. Other than that, calcium peroxide is classified as a kind of oxygen-releasing compounds, also called as ORC. The CP react in water to form H2O2 and CaOH2. In the application of calcium peroxide in water and soil treatment, several studies found that calcium peroxide is found to be a more effective source of hydrogen peroxide than liquid hydrogen peroxide for modified phenthane. As is well known, modified phenthane chemistry in subsurface involves the catalyst decomposition of hydrogen peroxide by iron 2 or iron 3 or native iron containing minerals to form hydroxyl radicals. They are very strong and relatively indiscriminate oxidants that can react with most contaminants at nearly diffusion limited rates. However, the instability of liquid hydrogen peroxide in the subsurface is the most serious limitation. Compared with hydrogen peroxide, calcium peroxide is relatively stable and can slowly release H2O2 by controlling its dissolution rate in water. Calcium peroxide can liberate a maximum of 0.47 gram hydrogen peroxide per gram of CaO2. It also can react for relatively long time and reduce disproportionate of H2O2 to oxygen. So these tables are the example of CP applications in contaminants removal in wastewater treatment process. So we can see that there are many types of contaminants such as glucose and glutamic acid, dyes, acid blues, glyphosate wastewater. Glyphosate wastewater, textile wastewater, bisphenol, toluic acid, methylene blue, and other material and other heavy metals. So such as in the red box is methylene blue, we can see that the action mode is photocatalytic degradation. The operation conditions is 0.01 millimole per liter, 1 gram per liter Cp under visible light. The removal efficiencies, the absorbance of methylene blue solution decreased significantly in 90 minutes. So the last part is the methodology. So the material that I use is 1 liter of methylene blue aqueous solution, 2 gram of calcium peroxide, 50 milliliter of buffer solution, 50 milliliter of aqueous solution containing copper, and the fifth distilled water. So the apparatus and instruments such as beaker, test tube, conical flask, measuring cylinder, and glass rod, and the last one is UV vis spectrophotometer. So these are the methodology are divided into 4 parts. The first step is sample preparation, second laboratory testing, and the third one samples analysis, the fourth data collection, and the last one results will be obtained. So during my preparation of methylene blue aqueous solution, 0.0372 gram of methylene blue will be dissolved in 100 milliliter of 250 milliliter conical flask. This solution was further diluted as desired. The solution is divided into various parts of beaker, and we will continue with the laboratory testing. So in the laboratory testing is to investigate the various effects on degradation of methylene blue. The first one is the effect of initial dye concentration. So under this part, 500 milliliter of methylene blue solution were prepared at 4 different concentrations, which are 100, 200, 300, and 400 milligram per liter. Next, 2 gram of calcium peroxide is dissolved in 20 milliliter of water and added into the methylene blue solution. And then the mixture was stirred and left for 3 hours at room temperature. At 1 hour interval, an aqueous solution of methylene blue was taken in a cuvette, and the degradation of methylene blue was monitored under the UV visible spectrophotometer. So the next parameter is the effect of pH. So the first step is the methylene blue solution concentration was prepared at the concentration of 100 milligram per liter, and then 2 gram of calcium peroxide was added into the solution. The solution pH was adjusted at 6, 7, 8 and 9 by addition of sodium hydroxide and hydrochloric acid. The pH of the methylene blue solution was maintained with 0.1 and sodium hydroxide to check the effects of different pH levels on the methylene blue degradation. The last one, the pH meter was used to measure the solution pH. The sample absorbance was determined using the spectrophotometer. And the last parameter is to be investigated is the effect of temperature. The degradation of methylene blue at first was studied by ranging the reaction temperature, and then 2 grams of calcium peroxide was added to the 3 samples of 100 ml of methylene blue. The next one, the degradation of methylene blue was observed at different temperatures of 30 degrees Celsius, 40 degrees Celsius and 50 degrees Celsius. After the desired temperature have been reached for each sample, 2 ml of each sample was taken into a cuvette. And then the last one, the sample absorbance was determined by using the spectrophotometer. So we studied in the various parameters is in order to find the optimum condition for the degradation of methylene blue using calcium peroxide. And then during the sample analysis, the spectrophotometer was used to analyze the absorbance at 663 nanometer before and after each experiment to measure the concentration of aqueous solution. And then the pH meter was used to determine the pH of aqueous solution. And the next one, the degradation efficiency is then calculated and the removal rates of chemical oxygen demand are studied. And then the data collection is obtained. The degradation efficiency for each sample was calculated from the following equation, which is the residual, the initial concentration of methylene blue is minus by the residual concentration and times by 100 to get the degradation of efficiency of methylene blue in percent. So that is all from me. Thank you.